Okay. Wow. Okay. Just take that one over. I'll pass this microphone over to the panel. Thank you.
Thanks. That's great talks, everyone. I've got one really sciencey question for Kitch, and then a broader question for this group. Think about standardization. Kitch, it was great seeing the single cell RNA-seq data with the cardiac tissues, especially. Thinking about that and the interplay between different cell types driving disease, have you guys considered omics-based or CRISPR pooled screens to look at what individual subtypes are driving phenotype? I was especially thinking about your cardiomyocyte single cell type not showing phenotype, but when you mix 9 cell types, you see a very striking phenotype.
You're thinking about Perturb-seq and
Perturb-seq.
Yeah, we'd love to do it. It's big money for a small startup, but yes, we'd love to do it. High throughput CRISPR knockouts of 25,000 genes, seeing what each one does in the different subcellular types. My ultimate goal, like a pathologist, was to have single cell RNA-seq as our disease readout for a lot of these organoids. It just comes down to cost right now.
There are some newer ways you could think about the inducible Cas system in your IPS that you then turn on once you're in your organoid stage.
Absolutely.
Throw your pool in, do a crop seq and look at.
We'd love to do that. Yeah, absolutely.
And then broader, everyone mentioned standardization, scalability, reproducibility. Leveraging AI and thinking about all these machine learning techniques, we need to have some sort of commonality to start pooling data together from multiple different platforms. What do you think about the scientific industry needing to do differently so that we have shareable, combinable data that we can start to identify patterns? And rare is no longer rare. Rare is just a different way to get to 5, 6, 8, 10 phenotypes.
I think you put the finger on the words sharing data. I think that will be the main issue. That is already a big issue to share data inside a single community of rare disease. So I have difficulty imagining how we are going to do it in between different community. Maybe there will be less competition between different rare disease, so the will to share will be bigger. But that's it's not only from the scientists that we have issue sharing data. It's more on the side of the IP and all the legal aspect and the legal team in academia, university, or I think it's also an aspect in CRO about sharing data. Then you decrease your IP. So that's I think the next challenge, and I have no idea how to solve it.
I can add a little bit because at the Children's Tumor Foundation, we actually have a data portal hosted by Synapse. And so over the last, I guess, 10 years or so, we've been first encouraging researchers to share their data, funded data, back into one central repository. But more recently, we're demanding it. So in recent years, it's part of our funding kind of award letter where researchers need to share their data back. And we have it in a portal. But to take it one step further is, now we need to share it outside of CTF. And so we have initiatives where we're identifying complete data sets that are shareable and putting them on public platforms like CBio portal is one of them we're working on, maybe Geo at some point. So we first have to identify which data sets are well annotated and formatted and go through all of that work, and then identify which public platforms to share them on. And we're actively doing that now. So.
I'll just second that, making it mandatory. Like I've worked with a number of research groups that said they didn't want to share the data. And I said, that's great, but then you're not going to get tissues to us. So that changed their mind, and every single one of them said, okay, we'll work with you. I think also patients are key here. Patients understanding the value of their data, patients getting the data if you can't get it, directly from the researcher, and patients starting to talk to people about, I want my data shared, I think is really key.
Yeah, second all that. In academics, you have to there's geo and all the repositories for seek data, but industry side, it's a mess to to keep all this data proprietary. And then, you know, our business model is partnering with pharma, who themselves don't want to share that data because also the IPS cells themselves, there's incredible royalties and fee structures and proprietary. You can't even screen for molecules without paying Japanese academia for the, you know, IPS academia for these things. It's onerous, the amount of boundaries that they place around IPS cells. So, you know, what we've got a workaround solution. We're able to create our own disease lines and then generate data from those. I would love to put that data somewhere in the cloud that's accessible to all. Right now, it's accessible to only our partners, which is unfortunate. But yeah, there does need to be some sort of industry-wide paradigm for addressing that.
Okay, I have a question, Kitch. So you talked a little bit about this, your 2.5 versus 3D organoids, but did you see any differences? Have you ever done experiments that shows really the outcome or phenotypes? Is what you see only possible with the 2D versus the 3D?
2.5D versus 3D.
2.5D.
I can only tell the practical differences we've seen. You know, they're relatively flat and they're attached. The 3D, they are floating in media. They are not able to get oxygen and nutrients into that central core for those spheroid type things. And so many of them get hollowed out, which can be a challenge with those. And then imaging, meaning that, you know, you're taking serial imaging of these things, because those spheroids are floating in media, each day that you're taking an image of them, it could be in this orientation, it could be in that orientation. So comparing it to between days is also a challenge.
Yeah, no, I get it, but there are some people that kind of optimize, standardized to a little bit at least, organoids when they're free floating, right? So did anybody do comparative studies between what you do and what?
I don't know that. Yeah, I'm not aware of anybody who's done actual comparison of the 2D versus 3D, other than the diversity of cell types seem to be the same in the 2.5D versus the 3D. You have smooth muscle cells, endothelial cells, fibroblasts in relatively the same quantities as what you'd see in 3D. They all beat. I showed you a lot of the beating. So beyond those metrics, you know, are they more or less mature? How do you define maturity? We can let these culture for 2-3 months if we had to. I'm quite confident they would be similar to the spheroids themselves, which can be matured as well. I honestly don't see a lot of difference, but if there's been a statistical quantitative study of the difference between the two, I'm not aware of one yet.
So I have a Okay, I wanted to ask 2 questions. I guess I'm gonna ask one only. Okay, I'll ask the second one later then. I have a question for the NF1 and the Armile, no, that you show that there is a lot of heterogeneity on both those diseases, no? In the case of the NF1, you show that there is so much phenotypic, no? I mean, in the symptoms of the people and also for the SMAD one, for the TGF beta. So do you think that's going to be make more difficult to find like a common therapy? Or do you think that we're going to need to find different therapies, I mean, kind of subcategorize inside of the disease, no? Different symptoms. And then do you think that we are going to need to find different therapies like small molecules or whatever for different these categories? Or do you think that it should be able to find something that encompasses the whole disease?
I think you're absolutely right. It's very heterogeneous with a broad spectrum of manifestations. So it will take multiple therapies and mechanisms. You know, we're just talking about NF1 today, but there's a whole other schwannomatosis type that I didn't have time to get into today, but absolutely, it's going to take more than one kind of therapy and more than one mechanism. Ultimately, combination therapies are something that are being looked at. So I think a combination therapy is on the horizon as well, to be able to hit multiple targets of the disease.
Yeah, I think it depends what you target. For us, if you target the SMAD4 or SMAD complex, then you can imagine that whatever the cofactor is next to it, you are going to act on all the different phenotypes. In our pipeline, we foresee that maybe we won't be able to do that. So we are also developing with the Geneton a FAB-CAR-T, especially for the fibrosis, in case we cannot get to the development of the small molecules. But since we start our first screening on really the destabilization of the complex, then the only limitation would be the delivery of the molecules, if it goes through the BBB or not. But if we would go for other like ALK inhibitors, for example, ALK5 inhibitors, that we also want to test, that would be only a part of the phenotypes. But going really at the origin of the issue, I think will allow us to go larger in the targeting of the phenotype. It depends where you go in the signal transduction. And since SMAD4 is really at the end of everything, then if we go for that, we hope that we will be able to tackle many different aspects.
Okay, thank you. I'm just gonna, one quick question from the online team, and then I'll get you all to lunch. Kitch, there was a question in regards to cost. How does the cost compare for the 2.5D versus typical cell and animal models?
Did you say cast?
Cost.
Oh, cost. And between that and animal models? Oh, that's an easy one. They're far cheaper. They're each organoid is less than $5, excuse me, to produce. And you can produce these, you know, in the thousands. So you can imagine the scale that would be impossible with these sort of things. So yeah, they're quite a bit cheaper.
Wonderful. That's great news, because drives the economics of the screen.
Indeed.
So again, I just want to thank all 4 presenters for a wonderful session. Thank you all for your attention and questions. Again, round of applause for our presenters.